Hi, students. We're going to cover a large diversity of life here. We're going to cover virus, bacteria, and protists. And even with this video, I expect you to still be reading your chapters. So, viruses are not going to be included in the classification of organisms. They're not considered a bacteria, archaea, or eukaryote. They are non cellular. They are considered to be obligate intracellular parasites. And scientists can't ever decide, are they really classified as living or are they not alive? So there are going to be two parts to every virus. The first part is they're going to have an outer capsid composed of protein subunits with an inner core, either with DNA or RNA, but not both. They won't have both. So notice all viruses have those two things. Now, some viruses will have spikes for attachment to their host cell, and they will also have an outer envelope. So, if you have a question that says a characteristic only found in some viruses, that would be your outer envelope. So, looking at viral reproduction is specific to a particular host cell. So, there's going to be a glycoprotein on the viral capsid is going to have a specific match to a receptor on the surface of a compatible host cell. So like a human cell, it's going to have, if you notice over here on the outside, how all these proteins are going to match up. They're going to have to match up perfectly. So they have to have they're specific to a particular host cell. So once inside the host cell, the viral genome takes over the metabolic machinery of the host cell and it starts creating more viruses. So this is going to be, we're going to use an example of a bacteriophage. And what that is, that's a virus that infects a bacteria. So there's two different cycles every time it reproduces. There's a lytic cycle and there's a lysogenic cycle. So the lytic cycle is the virus takes over immediately. The bacteria dies immediately after the virus is released. There's 5 stages in there. There's attachment, penetration, biosynthesis, maturation, and release. Then there's a second phase, the lysogenic cycle. So this is where it's going to lie, the virus or the phage in there is going to lie latent until triggered to enter the lytic cycle. And so that means it's dormant. It's not doing anything. The bacteria doesn't die until that phage goes back into that lytic cycle. So if we look at this, it's going to go through the lytic and then the lysogenic cycle. Now, this is a bacteriophage, but this is also pretty much how it works with a virus entering a human cell or a plant cell also. So the first thing, if you notice here, is attachment. Number 1, attachment. You need to know this. It's in yellow. With attachment, the virus attached to receptors. The second thing is penetration. The host cell takes in the entire virus or just even part of the genetic material. Then we move on to our third, which is biosynthesis, and that's where the host cell replicates the viral DNA. Then we move to the 4th stage of maturation, where the components of the virus are packaged into new viral products or particles. And then we move to the 5th stage, where the virus exits the host cell. It's going to burst through that cell membrane. And then when it does that, it damages the cell and then the cell dies. And so you can see with this circle here, we can go through first the lytic cycle, and then we can come back around and we can break into the lysogenic cycle. There's also plant viruses, and they tend to enter through damaged tissue and they move through the plasmodesmatin. Here's a good example is like a tobacco mosaic virus. And until recently, the only way to control viruses was to destroy the symptomatic plant. So if it had a virus, they would have to destroy this part of the plant or the entire plant. And then we would have to also control the insect vector if that is what is bringing the virus in. There's the animal viruses. They reproduce similar to bacteriophages. Some, but not all, will have that outer membrane envelope beyond a capsid. Animal viruses that have an envelope enter cells by either fusing with the plasma membrane or through endocytosis. Things like herpes virus, those are the ones that cause cold sores, genital herpes, and chickenpox, are infections that remain latent most of the time. So that means they're dormant, they're not doing anything. But when the human body goes through a stress, that can cause them to enter the lytic cycle, because at that point your immune system's not working as well. There's also other animal viruses that we're pretty familiar with, like measles is one of the most contagious human diseases. There's herpes simplex virus 1, which is associated with cold sores and fever blisters around the mouth. They say studies show that 70 to 90% of the adult population is infected. And then there's also the herpes infection of genitals, which is caused by the HSV2, and it's transmitted through sexual contact. And it affects 1 out of every 6 people in the U.S. population. Then we also have the varicella zoster virus. It's also a herpes virus, but that causes chickenpox. And later on, that can reemerge later in life as shingles. So antibiotics will not work against viruses. Immunization can help prevent infection by specific viruses. And then we can also use antiviral drugs, but not antibiotics. Antiviral drugs will target specific aspects of that viral life cycle. When we look at emerging diseases, these occur for the first time in humans and rapidly increase or enter the new geographic regions. Some examples that you need to know of emerging diseases are the H5N1, swine flu, HIV, West Nile, your MERS, Zika virus, and your SARS. The way that they emerge is a lot of times from they're transported to a new location, because we have boats that travel overseas, we have airplanes that travel overseas. They are able to infect new species, and then they have a new mode of transmission. They have very high mutation rates, and that enables the virus to infect new species. A viroid is a little bit different than a virus. It is naked strands of RNA not covered by a capsid. It's like a virus because it takes over the cell to make more viroids. And there's about a dozen crop diseases that occur. So the structural difference between a virus and a viroid is that a viroid does not have a capsid covering. Then if we look at prions, those are built from proteins and proteinaceous infectious particles. So what it is, it's a misshapen protein will change a normal protein into a more misshapen protein. So scientists discovered that this was the cause of Kuru and also mad cow disease. Now, viroids and prions are both alike, and that both of them are infectious molecules.
Kuru is a rare, incurable, and fatal neurodegenerative disorder that was formerly common among the 4 people of Papua New Guinea. Kuru is a form of transmissible spongiform encephalopathy caused by the transmission of abnormally folded proteins, which leads to symptoms such as tremors and loss of coordination from neurodegeneration. The term Kuru derives from the 4-word kuria or guria, due to the body tremors, that are a classic symptom of the disease. Kuru itself means trembling. It is also known as the laughing sickness due to the pathologic bursts of laughter which are a symptom of the disease. It is now widely accepted that Kuru was transmitted among members of the 4 tribe of Papua New Guinea via funerary cannibalism. Deceased family members were traditionally cooked and eaten, which was thought to help free the spirit of the dead. Women and children usually consumed the brain, the organ in which infectious prions were most concentrated, thus allowing for transmission of Kuru. Thank you for watching.
So if we look at the definition kind of prokaryotes, that means that there is no nucleus to contain any genome in there. There's no sealed compartments. There's no membrane-bound organelles. And we know that that means that there is not a membrane-bound nucleus. So when we're looking at prokaryotes, we're talking about bacteria and archaea. So the first living cells were going to be prokaryotes, and they were found in rocks about 3.5 billion years ago. The conditions on Earth very different from back then as they are today. There were really high temperatures. There was little free oxygen. Bacteria are going to be your most diverse and prevalent organisms on Earth. There's tens of thousands of different bacteria identified, and probably they will find that there's likely more than that exists. So if we look at the structure of them, there's three common shapes. There's rods, which are bacilla. There's spheres that are round, those are cocci, and then there's spirals, which will be spirilla or spirochetes. Some can form chains or bunches. Bacteria have no nucleus, and they have single circular chromosome found in the nucleoid region. I want you to pay special attention to that. It's not a nucleus, but it is found in the nucleoid region of the cell. Now, if we look at our shapes of bacteria, so if it is a round single bacteria, that is a cocci shape. And you're going to have to be able to identify and know the definitions of these shapes. A bacilla is a rod shape or pill. I always think that they look like those Mike and Ike candies, a pill shape. They can also be categorized by their arrangement of how they group together. So if we look at the prefix of a bacteria, like of the actual name of a bacteria, we can find that strepto means a long chain. So streptococci means chains of round shaped bacteria. And so if you notice the picture over here, it's a long chain of round bacteria grouped together. Then staphylo prefix means clusters. So if you hear the term staphylococci, that's going to mean clusters of round shaped bacteria grouping up together. Now, bacteria do have ribosomes, but no membrane bound organelles. So the outer wall is strengthened by a peptoglycan Bacteria do have ribosomes, but they do not have any other membrane-bound organelles. And we know that they have a nucleoid region, but not a nucleus. The outer cell wall is strengthened by peptoglycan, and that helps prevent it bursting or collapsing. And they may have an additional capsule outside the cell wall that will allow the bacteria to form what's known as a biofilm. Bacteria and archaea both reproduce asexually by binary fission. And so what happens if you look at the diagram over here? This is a better diagram right here. The bacteria copy the genetic material. It forms this cleavage furrow, which makes like a dent in the middle. And then it splits into two independent bacteria, which are both capable of growing into new individual. Binary fission is going to be quick and simple way for bacteria to make copies of themselves, and that's why they multiply very quickly. If we look at relationships of bacteria, there's heterotrophic, and they may be free living, or they may have symbiotic relationships with, you have commensalism, where one partner benefits but the other is not harmed or benefited. There's mutualism, where both partners benefit. So that would be like bacteria in your human intestines that helps make vitamins. So it's benefiting the bacteria and it's benefiting the human. And parasitism, which one partner benefits but the other is harmed. So that is going to be like our pathogenic or disease-causing bacteria. If we didn't have decomposers, life would halt. So decomposers digest dead organic remains and turn it back into inorganic nutrients to producers. And this leads into like the nitrogen cycle. And because plants are unable to fix nitrogen gas in the atmosphere, but they have to have nitrogen. And so bacteria are able to fix that nitrogen gas into a form that plants can use. And that's going to be a mutualistic bacteria, lives inside root nodules. So if we look at these legumes, and if you look at the the image over here, you can see this is the root, and then you see these round nodules on here. And so if we look at kind of how can we apply this? So if we take a large sample of soil, we divide it into individual pots, then separate the pots into two groups. The first group of every pot of soil has been sterilized. Then the second group, none of the pots of soil have been sterilized. So if you plant soybean seeds in the pots, what would you expect to see the seeds sprout and grow? So let's think this through. If the soil has been sterilized, then that means that there is no bacteria that the plants will need. Therefore, the plants in the non-sterile soil will grow a lot bigger because of the mutualistic relationship with nitrogen-fixing bacteria. We also have bacteria that is useful in our environment through bioremediation. So that's the ability of a bacteria to break down pollutants. And some of them are bioengineered, and then we've used them. We have used them in the past on examples of like when we had the Deepwater Horizon spill and other oil cleanups that we need. There is bacteria that will kind of consume and eat the oil.
The species we study is called smooth cordgrass, and it's the foundational species along the coast of the northern Gulf of Mexico and along the Atlantic coast. This species is incredibly important for building land and keeping land from falling into the sea. We went out to several sites that received heavy oiling after the spill. We found that the bacterial community inside the tissues of plants shifted to incorporate more bacteria that are important for breaking down oil. This is also really important because our collaborators have found that there's also oil inside of the plant tissues that still needs to be broken down. So it's possible that these bacterial shifts are going to be important for continuing to clean up the oil spill inside of the plant.
This is Bay Jimmy. It was one of the worst impacted areas after the Deepwater Horizon spill, and we used the shoreline to test different strategies for remediating the oil, for removing it from the surface. And after remediation, I went in and planted Spartina along the shoreline in different plots to compare what rates of erosion were like using different planted varieties versus leaving them unplanted after remediation. This stake right here is the back of a plot that we set up in 2011 where we were comparing planting versus not planting, and the plot extended 15 feet towards the water, and then in front of the plot was an additional 15 feet, and all of that's gone now. So since 2011, we have 30 feet of erosion up until this stake. Aspect of Spartina alterniflora is it's a very powerful ecosystem engineer, which means that it physically impacts this entire ecosystem. So just the presence of the marsh here is dependent on the ability of Spartina to hold the substrate in place.
The elegance of focusing on Spartina is that whether we're here in the Gulf or on the Atlantic or on the Pacific, there are Spartina everywhere. Some Spartina have been artificially introduced and have proven to be problematic, but the native Spartina and other congeners and related species that serve a similar function, that effectively engineer coastlines, can serve a similar purpose.
So now we're taking a closer look at these bacterial communities living symbiotically inside of root tissue to see if we can identify which bacteria are the best at biodegrading oil inside of plant tissue. And what we'd like to do is identify the best bacteria that we can use to inoculate plants in the future that are going to be used for restoration purposes. So instead of just putting a plant out there, we're going to put out a plant bacterial combination. And that combination of organisms is going to help clean up oil faster.
So pathogens are going to be able to produce a toxin and or adhere to surfaces and sometimes invade organs or cells. And so we know that we always hear bacterial diseases in humans. And so often toxins cause more problems than the growth of the microbe itself. So if we look at Clostridium tetani, which is called what we call tetanus or lockjaw, this bacteria actually lives in the soil and thrives in a puncture wound. The bacteria never leaves sight of the injury, but it releases toxins and it circulates in the blood. And those toxins are what prevent muscle relaxation. And where what eventually happens is the person suffocates when the diaphragm can no longer relax. So antibiotics generally either inhibit protein synthesis or inhibit cell wall production. Historically, the domain Eukarya was divided into four kingdoms: Protista, fungi, plantae, and animalia. Eukaryotes are now going to be divided into 6 supergroups in there, and protists are found in all of those supergroups. Now, we're not going to go through every one of the 6 supergroups. I'm just going to touch on several of them. So most protists are going to be single-celled eukaryotic microbes. So what does that mean? They have to have a membrane-bound nucleus. So some exist as colonies of cells, or some of them are multicellular, some are single cellular, but they are extremely diverse. So you need to know that protists are extremely diverse. So the best definition of a protist is any eukaryotic organism that is not a multicellular plant, animal, or fungus. Then if they're not a plant, animal, or fungus, then they're going to be a protist. Now, there's different classifications because they have combinations of characteristics not seen in any other eukaryotic groups. So traditionally, they're grouped based on the source of their energy and their nutrients. And this one we really want to look at is algae. We're going to talk about because they are photosynthetic and they make their own food like plants, but they're also classified as a protist. Now, here's our super groups in here, and we're not going to cover all these. We're just going to cover a few of them. And the first one that I think we're going to look at are the archaeplastids, and those are going to be your plants as well as green and red algae. So they are photosynthetic organisms. That means they make their own food through light, just like plants do. So this is going to be green and red algae, and they have chloroplasts, just like plants do. So basically, the algae are plant-like protists. They may be aquatic, living in the water, and part of the phytoplankton. They can be found on land, which they would be terrestrial algae, found in soil, rock, and in trees. Now, algae were traditionally classified based on the color of the chloroplast pigments. So there's green, red, and brown algae, and that's basically how they're classified. Now, paramecium is a ciliate, and it is a type of complex protozoan that moves by cilia. And those are the fine little hair projections on the outside of it. Now, another supergroup is where the amoebas fall into, and they move by pseudopods, which are processes that form when cytoplasm streams out into a particular direction. So they really lack a defined shape. So basically, what we think of an amoeba. And they feed by engulfing their prey with those pseudopods. Their prey might be algae, bacteria, or any other protist. The supergroup of choanoflagellates are flagellated protists that are most closely related to the animal kingdom. Protists have different types of locomotion. They have 3 kinds. They have pseudopods, just like the amoeba. Then the paramecium has cilia, those fine little feather-like projection hairs on the outside of it that help them move. And then if you look at this third one, you have a flagella, and that is an example of a euglena, and that flagellum tail helps them move.